Hello, everyone. Welcome to the Emerging Trends in Pharmaceutical and Biopharmaceutical Analysis. I'm Jerome Workman, the Executive Editor of Spectroscopy, and I'll be your moderator for this event. We are pleased to bring you this session, which is part of a two-day virtual symposium presented by Spectroscopy, the Society for Applied Spectroscopy, and the Koblenz Society. The Society for Applied Spectroscopy, or SAS, is a nonprofit organization dedicated to the dissemination of information related to spectroscopy. SAS is pleased to be the publisher of the internationally recognized peer-reviewed journal Applied Spectroscopy. The Koblenz Society is a nonprofit organization whose purpose is to foster the understanding and application of vibrational spectroscopy. Both societies have an educational mission, and this event is part of that mission. We invite you to visit both societies' websites to learn more. This virtual symposium is sponsored by CEM, PerkinElmer, Thermo Fisher, and Metrome, and Endress and Hauser. And now a statement from our sponsors. Today's event would not be possible without the support of our sponsors. CEM Corporation began manufacturing analytical instruments in 1978. In 1985, CEM developed the first microwave digestion system, revolutionizing sample preparation for trace metals analysis. Since then, continuous innovation has kept CEM at the forefront of sample preparation. With worldwide headquarters located just outside of Charlotte, North Carolina, CEM operates 7 subsidiaries as well as a large dealer network covering the globe. CEM truly is the global leader in microwave sample preparation. At Metrome USA, we're more than a leader in chemical analysis. We're a community driven by passion, integrity, and a commitment to quality and innovation. With expertise in both laboratory and process systems for titration, ion chromatography, electrochemistry, and spectroscopy, our approach goes far beyond just technology. Discover how our dedication to both science and humanity shapes everything we do at www.metrome.com. Customers worldwide trust Thermo Fisher tools and solutions to help them accelerate innovation and enhance productivity. Together, they are making advancements that truly matter. Thermo Fisher achieves this by providing an unmatched combination of innovative technologies, purchasing convenience, and comprehensive support. PerkinElmer is a global provider of scientific solutions with an 80-plus year history of bringing expertise, innovation, and technology to our customers. They manufacture the latest in analytical instrumentation and provide essential lab support services to streamline workflows. Partnering with scientists, clinicians, and lab technicians, they accelerate scientific outcomes and aid the development of groundbreaking products. PerkinElmer believes in the power of science towards a healthier, safer, and more sustainable world. Endress and Hauser provides advanced measurement and automation solutions for industrial processes across the globe. Their offerings include instruments for flow, level, pressure, temperature, and liquid and gas analysis. With a focus on innovation, reliability, and digital integration, they serve a plethora of industries. Their portfolio comprises of many products for any application, from basic to highly specialized, ensuring efficient and sustainable process control. We have a few housekeeping announcements before we begin. First, we encourage you to go to the resource section of our platform on the bottom of the media player. There you can find resources from our sponsors, as well as a link to view the other sessions of this event. We also encourage you to ask questions during the event today. You can submit questions by typing them in the Q&A box, which can be found underneath the media player. We will answer all questions at the end of today's event, after all talks have been presented. So please include the name of the speaker your question is for. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. We have a great lineup today, and I think you're going to enjoy hearing from all of our speakers. Today's morning session will feature talks by Igor Lednev, Joseph P. Smith, John Wozelick, and Julio Chacon Torres. I'm now pleased to introduce our first speaker, Igor Lednev. Igor Lednev is Williams Raycliffe Professor in Chemistry and SUNY Distinguished Professor at the University of Albany State University of New York. He is a co-founder of 2 startup companies commercializing a universal method for identification of body fluid traces for forensic purposes and screening tests for early diagnostics of Alzheimer's disease. Lednev is a founding director of the Center for Biophotonics Technology and Artificial Intelligence. He serves as an advisory member on the White House Subcommittee for Forensic Science. Lednev has co-authored over 280 publications in peer-reviewed journals and 10 patents, reaching an age index of 80. He received several prestigious awards, including the 2024 EAS Award for Outstanding Achievements in Vibrational Spectroscopy, the Charles Mann Award for Applied Raman Spectroscopy, and the Gold Medal Award from the Society for Applied Spectroscopy. Igor, thank you for joining us today. Please get us started.
Ladies and gentlemen, I'm Igor Lednev, Distinguished Professor and Director of the Center for Biophotonic Technology and Artificial Intelligence at University of Albany, State University of New York. On behalf of my collaborators, Drs. Lamia Al-Mehmadi, Sergei Verdata, Vladimir Yermolenkov, and Alexander Shechman, I'm very pleased to speak at this exciting symposium presented by Spectroscopy Magazine, the Koblenz Society, and the Society for Applied Spectroscopy. In this presentation, I would like to share with you our recent results of the study where we are trying to develop a new method based on ultraviolet Raman spectroscopy for in situ stability testing of mRNA vaccines. mRNA COVID-19 vaccines show a great potential of mRNA therapeutics in general. In 2023, Nobel Prize in Physiology or Medicine was given to Katalin Karika and Drew Weissman for their discoveries that enabled the development of effective mRNA vaccines against COVID-19. mRNA is inherently unstable, as we know, yet its stability is important for mRNA function. Lipid nanoparticles are used to protect mRNA in vaccines. In addition, mRNA COVID-19 vaccine manufacturers recommended low storage temperatures. Of course, we can expect that improper temperature control poses challenges, and as a result, we see the loss of approximately half of the vaccines distributed worldwide. Current methods used for testing the stability of mRNA vaccines are destructive. They are based on either gel electrophoresis or mRNA sequencing techniques. Both these approaches are labor-intensive, require special training and laboratory environment, and they are also resource and time-consuming. We believe that Raman spectroscopy can potentially provide automatic and in situ method for testing the mRNA vaccines. This approach could be non-destructive and label-free, quick, and potentially inexpensive. As you may already know, Raman spectroscopy is a type of vibrational spectroscopy. We use monochromatic laser light to irradiate the sample, and we collect inelastically scattered photons, analyze the energy, and build molecular vibrational signature of the material, which we can use either to elucidate the molecular structure or for material identification. Resonance enhancement of Raman signal occurs when the excitation wavelengths overlap with electronic transition in the molecule. The enhancement can be as high as 10 to 6, and this allows to get information from specific type of the molecule if we tune the excitation to the absorption of a specific chromophore. For example, in the case of protein myoglobin, excitation with visible light results in resonance Raman spectrum dominated by contribution from the heme. At 230 nanometers, the Raman spectrum is dominated by contribution from aromatic amino acids such as tyrosine and tryptophan, and when excitation goes close to 200 nanometers, amide chromophore from polypeptide bond dominate the spectrum. In addition, when deep ultraviolet light is used for generating Raman signal, the Raman radiation is moved into spectral range where there is no autofluorescence, meaning that there is no fluorescence interference. As a result, signal-to-noise ratio becomes high, and this results in higher specificity and sensitivity of Raman spectroscopy. A model mRNA vaccine in this study we prepared by mixing 250 microgram per ml of methyl pseudouridine modified luciferase mRNA and 200 microliter per ml of lipofectamine in the sodium herpes buffer. We use 266 nanometer excitation to generate resonance Raman spectra from mRNA. We use two approaches to degrade mRNA. In first approach, we added RNase A to the model vaccine. In second approach, we left model vaccine to degrade at room temperature and tested it at different time points. To evaluate the mRNA degradation, we used cell transfections and gel electrophoresis. In figure A, you can see the loss of the ability to produce a functional enzyme by luciferase mRNA due to aging. And on panel B, you can see the RNA gel loaded with equal amounts of lipid nanoparticles of different age. And finally, the panel C shows the quantification of the stained mRNA in the gel, which is shown in panel B. Let us discuss now the results of using deep ultraviolet Raman spectroscopy for testing the model mRNA vaccine. In this figure A, you can see the Raman spectrum of mRNA vaccine model in blue and the spectrum of lipofectamine lipids in buffer. It is clear that 266 nanometer excitation Raman spectrum of the model vaccine is dominated by contribution from mRNA. By the way, the peak at about 1000 wave numbers is from sulfate, which we used as internal standard. And it is also evident from this figure A that the 266 nanometer excitation Raman spectrum of RNA is dominated by contribution from ribose nucleotides. Based on literature, we were able to assign main Raman bands of mRNA to adenine, guanine, and cytosine. Also, I need to let you know that we are not able to assign each band to specific vibrational mode. In this figure, we illustrate the results of using Raman spectroscopy to probe mRNA degradation. In figure A, you can see Raman spectra acquired before and after RNase A addition. You can see significant decrease in Raman bands of mRNA. In the case of model vaccine aging, the decrease or any change in amplitude of mRNA Raman bands is not evident. However, in both cases, because of RNase A effect and aging, we see small but reproducible shift of two strong Raman bands, 1482 and 1580. The shift is evident, as I said, both due to addition of RNase A and due to aging experiment. And the shift is several wave numbers. It's not a big shift, but it is quite reproducible. And the error bars here represent the variations for three separate experiments when we prepare model vaccine when we prepared new model vaccines. Literature actually showed that the band at 1580 in RNA can shift to high wave numbers due to base unstacking. And you can see, for example, here how the shift occurs. And as concerns 1482 band, this also was known from literature that it shifts to high wave numbers due to interaction between lipids and nucleotides. So tentatively, we can assign this shift which we found in our system to the same phenomena. What is most important is that this change in band position for mRNA due to aging effect is qualitatively similar to the changes which we found due to cell transfections and the gel electrophoresis. In other words, it is consistent, the behavior, Raman spectroscopic behavior of these bands is consistent with a control experiment on degradation of mRNA. Well, as I indicated earlier, the shift of the Raman bands are relatively small but consistent, and in order to accurately determine the shift, we used multiple peak fitting of mRNA vaccine spectra, and we used the Gaussian shape for peak function. In our model mRNA vaccine, we used lipofectamine as a lipid which forms nanoparticles. And this is because we couldn't use lipids of real vaccine, for example, from Moderna. Although the composition of Moderna mRNA vaccine is well known, lipids are known. However, the protocol for forming lipid nanoparticles is proprietary. So what we have done in this experiment, we actually used real lipids from Moderna vaccine, and we measured deep UV Raman spectrum, and we found that the spectrum Raman signal is again weak, and we know that if we would add mRNA into this solution with these lipids, or if we would measure Raman from a real Moderna vaccine, the spectrum would be dominated by signal from mRNA. Well, I hope that the results which I shared with you convince you that deep UV resonance Raman spectroscopy has a great potential for assessing RNA stability in RNA vaccine in situ. When fully developed, the proposed method can be adopted to a portable instrument, which is actually commercially available already. Deep ultraviolet Raman instrument, both portable and stand desktop, are available currently. And deep UV Raman spectroscopy could characterize RNA at various manufacturing stages, including product quality control, for example, by probing mRNA integrity and stability, or optimizing storage conditions. As a future study, we are currently working on evaluating the distribution of mRNA between liquid nanoparticles and buffer using deep UV Raman spectroscopy. For further details, I would recommend you to look at our paper, and let me acknowledge my collaborators, Dr. Lamia Al-Mehmadi. This study was one of chapters in her PhD dissertation, and now she is postdoctoral fellow at MIT. Dr. Sergei Verdata, who is expert in molecular biology. He made model vaccines for this study, and he's working with Professor Alexander Shechman in his lab. And Dr. Vladimir Yermolenkov, who is expert in deep ultraviolet Raman spectroscopy, and he assisted us with experiments, and we are grateful to NIH for financial support, and thank you very much for your attention.
I'm now pleased to introduce our next speaker, Joseph P. Smith. Joseph P. Smith is currently director at Merck within the Data-Rich Experimentation Organization in the Process Research and Development Department. At Merck, he leads a multidisciplinary research group devoted to inventing, developing, and deploying data-rich technologies to enable process-focused efforts across small molecules, biologics, and vaccines. He earned his BS in chemistry from the Pennsylvania State University in 2012, where his research focused on synthesis, purification, and characterization of magnetic nanoparticles. Dr. Smith then earned his PhD in analytical chemistry from the University of Delaware in 2017, where his research focused on developing novel analytical tools leveraging spectroscopy, hyperspectral imaging, data science, and chemometrics to analyze geological and planetary materials. Dr. Smith then began his industrial career at Merck in 2017, where he remains today. He has published over 57 peer-reviewed articles to date and currently has over 1,565 citations with an H-index of 18 and an I-10 index of 30. He has received the Emerging Leader in Molecular Spectroscopy Award, the Bruce Kowalski Award, the Glenn Skinner Memorial Award, the Eastern Analytical Symposium Graduate Student Award, and the University of Delaware's Excellence in Teaching Award. Joseph, thank you for joining us today. Please get us started.
Hi, my name is Joe Smith. I'm currently a director at Merck in the Process Research and Development Organization. And today I'll be talking to you about the intersection of spectroscopy and data science, the next generation of enabling technologies for pharmaceutical process development. And so just to start, I'd like to give a really big shout out to the Process Research and Development external landing page. If you follow the QR code or the link below, you can learn more about the Process Research and Development Organization as a whole, including any open job postings, who we are as an organization, what we do, some really neat testimonials from different colleagues across the organization. So please feel free, follow the QR code, follow the link to learn more about Process Research and Development at Merck. And so just to start, I'd like to give a really huge shout out to the entire organization that I'm a part of, Process Research and Development Enabling Technologies, specifically within the Data-Rich Experimentation or DRE organization. All of the work that I'm going to showcase today couldn't have been done without virtually all of the help of the members of Process Research and Development Enabling Technologies, and specifically DRE. So I'd just like to thank everyone that's been a part of the work. And really start off with acknowledging everyone's contributions. And so with that, what is data-rich experimentation at Merck, and what is our mission and vision? So in short, our mission is to develop and deploy cutting-edge solutions to maximize the knowledge generated from every experiment conducted across process research and development at Merck. We strongly believe in innovation through partnership. So building those trusted partnerships and transformational relationships as we develop and deploy our technology is critical. Our vision, said briefly, is that we aspire to embed data-intense tools into the fabric of our organization's process development culture, and really to drive transformative innovation to deliver business value through adoption and deployment of modern technologies. And so building a little bit more on that, from a data-rich experimentation point of view as our larger organization, and specifically where my group kind of fits into this, how do we go about impacting process research and development. And one of the really key parts of our groups is that we are a multidisciplinary cross-modality organization. So the team that I am lucky enough to lead supports activities across all of the different disciplines that are shown here. So we do a lot of data science, machine learning, artificial intelligence, modeling, simulations, a lot of data engineering work, systems and hardware integration, systems biology and multiomics, process analytical technology, which I'll talk a lot about today, kinetic studies, mechanism elucidation, high throughput experimentation, custom automation, and even more. So really multidisciplinary kind of cross-functional work that the group is focused on, with at its core maximizing the knowledge gained from every experiment across process research and development. And so how do we go about doing that? One of the core foundational principles for us is collaborating across modalities, functions, and sites. So our organization supports anything from traditional small molecules to monoclonal antibodies or antibody-drug conjugates, all the way up to vaccines and virus-like particles and mRNA lipid particles. The team that I lead is across West Point, Boston, and Rahway. And really, one of the pillar foundational principles for us is trying to establish cross-functional stakeholder partnerships. So we work across a variety of multidisciplinary teams from a variety of different organizations at Merck, and really, again, applying a lot of those disciplines to support all modalities across a variety of sites and a variety of different stakeholders and partners. And so diving deep into kind of one of the tools and technologies that we are invested in within data-rich experimentation and process research and development enabling technologies is called Process Analytical Technology, or PAT. And so for those that maybe aren't as familiar, what is PAT? PAT was defined formally by the FDA in 2004 as a mechanism to design, analyze, and control pharmaceutical processes. And so for us at Merck, PAT really offers the ability to collect data that's often considered inaccessible, to enable a deeper process understanding to facilitate best science at first filing. PAT offers the ability to provide real-time in situ monitoring of processes across small molecules, biologics, and vaccines. And it really enables a truly representative analysis for process scalability and robustness, also really terming for us the eyes into the process. And so when we go about developing and deploying PAT tools, how do we go about doing that? And so for us, said shortly, for sample collection, none is best. What that means is essentially there are 3 tiers of PAT tools that we like to define. Those are inline, online, or at-line. I can explain what that means. So essentially, as the figure depicts on the right side of the screen here, an inline tool is something that is directly interfacing with the process, measuring process phenomenon in situ in real time. And so an example of that might be a Raman probe sitting within a bioreactor. That would be inline. If you're not able to achieve inline technology development and deployment, we then typically go to online tools. So what that means is that we will need to flow the process to the analyzer, whatever that may be, and then we can either flow to waste or flow back to the process, as shown in the diagram here. If online tools are not available or possible, then we will sample the process and then measure that in an offline manner. And so if the offline analysis is close to the process, then that is typically termed at-line. If it is further away from the process, then that is typically termed offline. And so the reason why we prioritize inline to online to at-line or offline is really at the bottom left side of the screen here, where we've turned the observer effect to the sampler effect, in that the mere sampling of a phenomenon may and can inevitably change that phenomenon. We have some examples here that we want to showcase in terms of that happening. And so because of that, if we have an inline tool that sits directly within a process that provides analytical information about that process in situ, inline, that is preferable whenever possible. And so one of the first case studies that we wanted to share today was really around enabling vaccine process development using PAT and specifically in situ Raman spectroscopy with an emphasis on measuring and determining and quantitating nutrient and metabolites during an upstream vaccine process. And so as many folks know, Raman spectroscopy provides chemical and molecular information about a sample, and using an in situ probe-based Raman spectrometer is able to determine nutrients and metabolites across the biopharmaceutical process, i.e. a cell culture. When we put a Raman within a bioreactor, for instance, for an upstream vaccine process, we are collecting three-dimensional data. So we have our spectral range, we have our intensity or counts at each wave number, and then we have time as that third dimension. So we're able to collect really data-dense Raman spectra across the entirety of the process that we can then interrogate using a variety of data science tools to learn about the process, provide deeper process understanding, and provide the ability to, for instance, measure and monitor nutrients and metabolites during an upstream vaccine process. And so once we've gone about collecting those really dense Raman spectra, how do we go about analyzing that? So for us, data science is the key to unlocking the potential of PAT. And what we mean by that is essentially following the process that's shown on the slides here, where we acquire the Raman spectra, we then acquire reference Raman spectra for a variety of target analytes of interest. We then go through spectral preprocessing, variable selection, to be able to feed the most critical information about chemical and molecular phenomenon within the process to our multivariate chemometric approaches. So in our case, using multivariate partial least squares regression for building our model, and then evaluating our model and selecting the optimal model for, in our case, nutrient and metabolite analysis in real time. And so when we're studying an upstream vaccine process, what are we looking for? What are some of the interesting phenomenon from a pharmaceutical perspective in terms of the vaccine process development? So in our case, when we're looking at, in this example, a yeast fermentation for protein expression, there are three critical parts to that process. The first is the batch glucose phase, where we have initial buildup of biomass. The next is the exponential glucose feed, which we're further building biomass. And then kind of the next part is the induction of protein expression, where we're feeding or having a bolus addition of an inducer to, again, induce that kind of protein expression. Over the entirety of that process, we are hoping to analyze and our kind of target analytes of interest are glucose, which is our carbon source, our inducer for protein expression, which is a secondary carbon source that again enables the induction of protein expression, our ethanol, which can indicate shifts in metabolic states, and dry cell weight, which is essentially a net biomass production. And so now that we have kind of an understanding of the process and what we're hoping to measure using our inline Raman spectroscopy tools with a lot of our multivariate chemometrics PLS approaches, when we put that all together, we are indeed able to successfully monitor a variety of important critical process parameters and important features across the entirety of this process. So as you can see here, when we start with inoculation of the process and we have our batch glucose phase, we can see and monitor in real time glucose shown in blue there. We can also see our ethanol tracking metabolic shifts throughout the process. We can see as we are kind of inducing protein expression, we are able to monitor and measure the inducer consumption. And finally, across the entirety of the process, looking at dry cell weight, tracking with biomass all the way to cell harvest. So all in all, putting this technology together with Raman spectroscopy and a variety of data science approaches, we are able to now understand the process at a deeper level, and we're able to monitor and quantitate these four critical analytes of interest for the process itself. And so now that we have that technology available, how do we go about applying that further for vaccine process development? So one of the important steps across the pharmaceutical industry in large molecules is clonal selection for process development. So selecting an optimal clone to produce your therapeutic of interest or your protein of interest, for instance. So in our case, we applied this technology across clonal selection. And in our case, you can do that with having a variety of different clones, applying the same process condition, and understanding the process at a deeper level using this technology. You can also take different clones and apply different process conditions. You can also take the same clone and apply a variety of different process conditions. So in our case, all in all, this technology was used to directly influence decision making in terms of how we're selecting the optimal clone. And you can see from just looking at this slide, right, when we look at different clones at the same process condition, we can see different phenomena occurring. We can also see when we look from a clone one, process A and B, for instance, the left side of the screen, we can see definitely some stark differences in what is happening from a process perspective for those different clones, which again can help us make the best decision to produce the highest yield, highest purity vaccine process we can. And so now that we've applied that to a really important step for clonal selection, we can also further deploy and implement this for clinical supply studies and for commercial manufacturing processes. So this technology was indeed successfully deployed across clinical scale operations, across commercial manufacturing. And one of the case studies shown on this slide here showcases why that's critical. So we can help to kind of ensure consistent process performance using this in situ real-time methodology. We can also identify process excursions or deviations. So for instance, if we look at the right side of the screen here, if we have an inadvertent bolus of glucose due to an equipment malfunction, we can now see this in real time. We can act on this urgently. We can mitigate a variety of risks that are happening here. So not only does this provide us with a deeper level of understanding, again, a more eyes into the process, having a tool that directly interfaces with the process itself, providing real-time representative information about metabolites, nutrients, biomass. We can also use this further to make sure that the process is performing in a robust, consistent way and act on any deviations immediately as we see them. Next, kind of switching gears, moving from a kind of yeast fermentation process, can we also apply process analytical technology to live virus vaccine cell culture processes? And one of the important things to note that for live virus vaccines, typically there are a variety of biosafety considerations that limit our ability to sample the bioreactor. And so if this is a biosafety level 2 process, for instance, we really don't want to sample that bioreactor if we don't have to. And so how do we provide deeper understanding about a process when it might be difficult to actually sample and analyze? Well, inline PAT solutions are what we're proposing to help solve that challenge. And so one of the tools that we explored in this case was dielectric spectroscopy, also known as capacitance, as a PAT tool for determining and quantitating viable cell density and cell viability within a live virus vaccine program. Similar to Raman spectroscopy, dielectric spectroscopy can provide three-dimensional information shown at the bottom side of your screen here. So you have frequency, you have your capacitance signal, and then you have time in terms of the process. As we can see from there, we can see changes across that three-dimensional data set, which gives us confidence that we'll be able to explore this of, hey, if the capacitance signal is changing across the process, can we leverage those changes to understand and monitor critical process phenomenon? And said shortly, how dielectric spectroscopy works in this case is that viable cells with intact membranes are polarizable and thus contribute to the signal being measured, whereas dead cells with ruptured membranes fail to polarize and are not measured. So we can perhaps leverage this phenomenon to be able to determine cell viability and viable cell density, which are really important for developing a vaccine. And so, like I mentioned, when we apply this inline in situ dielectric spectroscopic technique, we're able to generate three-dimensional data shown here, which we can look at the full data set. We can zoom in on certain low frequency ranges, for instance, to provide a more detailed look at how the capacitance signal is changing over the course of time. And so we have really, really rich information within this three-dimensional data set. And so how do we go about extracting all useful chemical and molecular information from that data set? So first, we take an exploratory approach of using univariate analysis and looking at some canonical frequencies shown on the slide here of what is hypothesized to be able to determine important cell viability or viable cell density phenomenon. As you can see, univariate analysis in this case doesn't really cut it. So we have 136 kilovolts on the left side of the screen. We see some decent correlation with cell viability, for instance, but looking across the board at any one singular discrete frequency, we really don't see the ability to track viable cell density or cell viability in the way that we want to, as compared to kind of our offline reference standard shown in black. However, when we take a more data science and chemometric and multivariate partial least squares approach, we are indeed able to now to be able to, in this case, track and quantitate viable cell density across the process using kind of partial least squares in conjunction with dielectric spectroscopy, which offers a significant improvement over traditional approaches that leverage univariate linear regression, for instance, where again, we're seeing really good agreement between what we're measuring from an offline perspective and what we are predicting using this inline data science plus dielectric spectroscopy approach. And further, we see a very similar trend when it comes to cell viability. So we're offering significant improvements using this inline real-time PAT measurement, where we now see really good correlations between our predicted and measured, and specifically just calling this out in terms of those later process phenomenon where it's historically difficult to measure cell death. Again, we're able to now have an inline technology to be able to quantitate, monitor, and measure cell viability and viable cell density. And so kind of putting all that together, we're now able to apply and deploy this technology using data science, using dielectric spectroscopy hand in hand across 10 different independent live virus vaccine bioreactors to really be able to now quantitate cell viability and viable cell density in real time without the need to sample or interface with the bioreactor in a fully automated way, offering tremendous advantages, providing more data-rich information about the process, and also limiting any need to sample the bioreactor. And next, we wanted to ask the question, okay, dielectric spectroscopy worked. That was really informative for cell viability and viable cell density. But could we push our technology development even further and explore a lot of novel PAT approaches that have simply never been applied to a live virus vaccine process in general? Dielectric spectroscopy was one of those techniques, but we wanted to say, hey, there are more. So can we actually explore a lot of these PAT tools that, again, are all inline, so all directly interfacing with the process itself, ranging from Raman, from ATR FTR spectroscopy to inline microscopy and 2 different tools there, and also kind of particle size information from focused beam reflectance measurements. So all in all, essentially throwing the kitchen sink at this live virus process, for instance. And what we see here across a variety of the tools is really exciting. And so from our first in situ microscopy tool, we can see when we look at this microcarrier adherent cell culture live virus vaccine process, we can see that we are indeed able to observe a variety of different microcarriers and cells growing within them. We also see, using our kind of analogous in situ microscopy approach, which is taking a more zoomed out field of view, for instance. So with our previous in situ microscopy approach, we're really zooming in on those microcarriers, being able, like I mentioned, to see the adherent cell culture process, seeing cell growth within or on top of, the microcarrier itself. Using this in situ microscopy tool, we're able to have a more global and zoomed out approach where we can see multiple microcarriers offering kind of complementary information and learning more about the process itself. And really just to kind of like summarize that inline microscopy portion, and like I mentioned, linking back to that inline versus online versus at-line type of analysis. So in this case, inline PAT tools were allowing for direct visualization of the process in an in situ manner. That was really important because when we looked at this process historically using offline microscopy, we would sample the bioreactor, we would then perform offline microscopy, shown at the bottom left side of the screen here. And one of the things that we're looking for instance, is microcarrier clumps, so clumping of those. From the offline microscopy, we don't really see that too much. But as you've seen in the previous two slides and on this side as a summary, we do indeed see when we have an inline, truly representative analysis within the process itself, we do indeed see now microcarrier clumping. So again, highlighting the need for PATs as an eye into the process. We hypothesize that we don't observe this for the offline microscopy due to shearing during sampling of the bioreactor, again, linking to that kind of sampling effect that was mentioned earlier of the mere sampling of a phenomenon may inevitably change that, further pointing to the need for inline PAT tools for truly representative analysis. Then linking to that, we also explored Raman spectroscopy for live virus vaccine development. We were indeed able to quantitate 5 different critical nutrients and metabolites across that process. We were also able to use ATR, FTR spectroscopy similar to Raman, collected across the entirety of the process. And similar to Raman, we were able to indeed measure using ATR, FTR spectroscopy these 5 nutrients and metabolites, again, linking to the fact that we have diverse tools that can all be inline, all directly interfacing with the process. So if we have a difficult to measure analyte of interest, we have now a lot of optionality between can we use Raman, can we use FDR, for instance, to probe best the certain analytes of interest that we need to understand from a pharmaceutical perspective. We can also look at focus beam reflectance measurements, as mentioned earlier. And so here we're providing a 3-dimensional data set for cord length, time, and counts. And so similar to perhaps a particle size distribution, we have a cord length distribution here that we can now get information about sizing throughout our process. So you can see going from cell plant all the way through harvest, throughout the various portions of the process, we can see how our focus beam reflectance measurements are indeed changing, allowing the ability to monitor key aspects such as media exchange and infection all the way up to harvest, things of that nature. And just leaving with the last couple of slides here, when we're not able to develop and deploy an inline PAT tool for analysis of a pharmaceutical process, we invest heavily in the ability to automate the sampling to enable that. So just some of the recent work that we're doing now, leveraging commercial instrumentation in the SegFlow PS, and applying some custom automation to this commercial setting to be able to have automated liquid handling, to be able to have cell-free sampling and cell-containing sampling, and to be able to multiplex a lot of the sampling across a variety of different bioreactors to increase our data density, improve sampling robustness, reduce safety risks, and really help achieve a more lights out process operation using automated sampling. And so before the acknowledgements, I just want to really highlight that we're also invested in deeply advancing both Merck's pipeline and the field in general. We are always open to collaborate. We have a ton of collaborations ongoing. We really strive, again, to be able to impact the entirety of the scientific community, and the entire team has done an excellent job of looking at really cutting edge publications and trying to promote the science that we're doing to benefit the entirety of the scientific community. So if anyone is interested in collaborating, if anyone is interested in learning more, please reach out to me. We are always happy to collaborate and connect to try to advance the science in a partnership focused way. And with that, would just really like to thank everyone that's been a part of the work. Would not be possible without everyone, and probably more listed on this slide. And thank you all for your time.
Thank you, Joseph, for that excellent talk. I would now like to introduce our next speaker, John Wesolek. John Weselick, PhD, retired. He's a former scientific director at Bristol Myers Squibb Company with over 30 years of experience in analytical chemistry. He oversaw several analytical laboratories supporting various stages of pilot plant scale-up in both small molecule and fermentation. His focus has been on the application of inline and offline spectroscopy for the purpose of enhancing process knowledge and positively impacting the drive towards sustainability in pharma and biopharma arenas. Application specialist in process analytical technology for process control and understanding in small and large-scale reactors, foreign material investigations, reagent quality and stability, monoclonal antibody and antibody drug conjugate stability and processing. John, thank you for joining us today. You have the floor.
Hello, everyone. Thank you for joining in. Title of the talk is Taking Advantage of Multiple Tools in Pharma Biopharma Development Cycle. First, I'd like to begin by going over the 12 principles of green chemistry with regards to the type of analysis that we like to do. In terms of green chemistry or sustainability, we like to use safer solvents, have instruments that are designed for energy efficiency. We want to avoid waste as much as possible. And we want to be as safe and protect ourselves from accidents as much as possible. So these are some of the key drivers, as you can see here. The instruments we tend to focus on are spectroscopy based. We also use additional instruments such as particle analyzers and mass spectrometry. The advantage of using these tools is safety, speed, process knowledge, which is key for kinetic profiling. We like our methods to be non-invasive, rapid, easy to transfer, and of course, sustainable. Little or no sample preparation. Our green instrument capabilities, and this is a list, but it is not an exhaustive list. We use Raman, near-IR, IR, UV-Vis, also fluorescence, and mass spectrometry and particle analyzers, as I mentioned. They replace key analytical tools such as Carl Fisher, other titrations, replace GC, replace PXRD, replace HPLC wherever possible. What are our goals? As I mentioned, safety. We want simple, no sample preparation required is ideal, but otherwise we want to keep it simple. We like our methods to be rapid and non-invasive. We like instruments that are robust with low maintenance and minimal moving parts. We want to generate minimal or no waste whenever possible. We will use chemometrics, and we want to be able to transfer these methods from one instrument to another instrument with ease and reliability. An example here is Poloxamer 188. This is a stability and purity study. We used a number of tools listed here. We also exposed it to different conditions, environmental conditions. The question came up from our colleagues in the biopharma area was that they had a lot that they had trouble with. So we went through the protocol of exposing it to light, different air temperatures, different conditions, and we found really no change in the Poloxamer, in this case, Poloxamer 188. It seemed to be very stable under our conditions of stress. But unfortunately, sometimes reagent stability and quality, well, it's not exactly what I ordered. Here's an example of 3 lots of Poloxamer 188. Vendor 1 and 3 seem very similar. Vendor number 2 had a carbonyl stretch. It should not be there. We decided to investigate what was going on. So we already know that it's very stable under these conditions. We turned to one of our NMR specialists, and he did some nice NMR work focusing on C13. And what we found out is, yes, there were a high level of impurities. The impurities may have been from breakdown or contamination, but the polyoxyethylene, polyoxypropylene ratios were off. The molecular weight, total molecular weight, was off. So it suggests that what we had purchased was either contaminated or mishandled and broke down. The whole idea here is, can we make a quick assay, such as with FTIR, to determine the quality of the material so we know that we can proceed with some reliability. Another example here is in polyethylene glycols. We know that PEGs do break down and can form organic peroxides. We know that light can induce this breakdown. So we began to take a look at the breakdown based on a long-term stability study where they saw some unusual behavior in one of our drugs under investigation. We turned to AbChem peroxide test, which is a chromogenic test utilizing UV visible spectral range. We use that to check the quality of the material, and we used a solo VPE system so that we didn't have to work about we utilized Beer's-Lambert law, where we're able to change the path length and not have to deal with multiple dilutions in order to build a calibration curve. We then focused on the PEG in question, PEG 400. We decided to look at other polyethylene glycols, such as 200, 300, up to 1,000. We exposed it to high intensity, 18,000 lux of LED light under different conditions, such as inert atmosphere and in room air. And what we found was lower molecular weight PEGs formed organic peroxides at a much higher level than the more viscous, less oxygen permeable, higher molecular weight PEGs, as shown in the right. So what we found here, which we already knew that PEGs were light sensitive, and we knew that there was a potential for affecting product quality, especially if your active pharmaceutical ingredient is susceptible to organic peroxides. But we also found out that with lower molecular weight PEGs, they are more dangerous to our active pharmaceutical ingredients for those that are subjected to organic peroxides. We wanted to take another look at the breakdowns of PEG 400 under these lights. We turned to an NMR specialist, and he began taking a look at the proton NMR. Did some two-dimensional work, as shown here, and came up with a prediction of pathways based on what he was able to identify by NMR. For example, the presence of trace amounts of acetic acid, of formic acid, of PEG formate. So we know now much better what our breakdowns are of polyethylene glycol when subjected to intense light or normal light or over a long period of time. We also have utilized some vibrational techniques such as infrared spectroscopy in this particular case. And I will point out that many of these instruments I'm talking about, we utilize open access. So we build methods and train chemists so they can just walk up, put a sample on, and analyze their sample and get a rapid result. Shown here is in a biomedia preparation The complaint was that there was a precipitate being formed. They believed it was cystine. It impacted their biological material, their production material. So we began to take a look at what is the effect of copper, along with other trace metals, on key amino acids. So we took a number of key amino acids, added some metals such as copper, and during the preparation, in parallel, we subjected it to different conditions. We saw that if the copper was subjected to various pHs and mixing rates, and the protocol was not accurately followed, copper 1 would form copper 2, which led to the dimerization of cysteine to cystine. The cystine precipitates and is not utilized by the cells during production of a biological material. We also found trace amounts of phenylalanine. That is why we expanded the study to utilize other amino acids such as phenylalanine, histidine, tyrosine that may precipitate during media preparation if a careful, proper preparation is not followed. Another instrument we utilize is FTIR from BioTools. This instrument is able to take a raw spectra, we Fourier transform that spectra, and we take a look at the amide 1, amide 2 bonds after we've subtracted the buffer. And what we end up with is some very interesting information that can then be applied to a standard library that is applied by the company, so we could look at secondary and tertiary characteristics of the protein. First example here is BSA. This is the first step in understanding how we could utilize this tool. We know that BSA can form aggregates depending on the temperature. It can gel and you lose the activity. So we see changes in the amide 1 and especially broadening in the amide 2 region. We typically deconvolute the amide 1, amide 2 region, but we can also deconvolute the fingerprint region, as shown on the bottom. What we see with BSA, if we increase the temperature, we see a big change in the alpha helix, a big change in the beta sheath, and a big change in the coil. Lesser change, for example, in the turn as you're increasing the temperature. So let's take it another step. Now we take one of our proteins and we look at its biological activity coupled with its stress at increasing temperatures. And we know from this protein that it tends to gel around 65 to 80 degrees centigrade. If you do go higher in the order of 95, 100, 105 degrees, it tends to aggregate. I will point out that at up to 80 degrees, you can actually cool it down to ambient temperature, allow it to sit for an extended period of time, and it will reform to its natural state. But once it aggregates at a much higher temperature, it is permanent and you've lost all your biological activity. So taking that into account, we looked at the alpha helix as we increase the temperature. We looked at the coil. We see some changes in the sheath. But as a result, we reach a point of no return when we get to the higher temperatures. This gives us an indication of where we may be able to utilize this in a flow system during, let's say, tangential flow purification, where you want to avoid or limit your amount of aggregation. We can also take a look at the fingerprint region via Raman spectroscopy. You can see here we could focus on some of those key amino acids that are involved in aggregation: phenylalanine, tyrosine, tryptophan, for example, the sulfur-sulfur bond. We could take a look at those changes during temperature changes or changes in buffer concentration if you're doing a purification process. Here's just a quick example. This is what occurs during pre-aggregation. We're looking at 1623 reciprocal centimeters. We're looking at the phenylalanine shift, and this is all pre-aggregation. If we reach a point where it shifts enough, we're going to lead towards aggregation, and it becomes a predictable event. And this just shows you, once we go a little bit longer and analyze the data, we can look at how it correlates with van der Waals forces. Another instrument we utilized was HORIBA's A-team system, transmission, excitation, emission matrix. Our goal was, let's look at the stability of our MABs, and how can we apply this to commercial ADCs along with commercial MABs. So here's a monoclonal antibody using their parallel factor analysis. We get a nice spectra of the monoclonal antibody in its natural state. If we take a look at the ADC, antibody drug conjugate, in our particular case, we were actually able to differentiate between the monoclonal antibody, the linker, and the active pharmaceutical ingredient, or what I like to call the warhead. We could take that one step further and just do a kinetic study. Take it to 30 degrees, for example, for a period of 60 hours, and we can monitor live the breakdown or the deformation of the ADC, something that we want to avoid during our process purification. So what have we learned here? We could take a look at the UV region with the A-team fluorescence fingerprint. We can take a look at its aggregation rate, look at the linker, active ingredient, monoclonal antibody ratio to make sure that we're on target with our drug of design. And we can also take a look at the stability of monoclonal antibodies and antibody drug conjugates. It's another example, and this is going to be for both small and large molecules. The question is, to Carl Fisher or not to Carl Fisher? Carl Fisher, reagents are toxic. They generate waste. There's a lot of sample handling. It's invasive. If we use near IR, there's no waste, no sample handling, non-invasive. So to really understand this, we first did some studies with Carl Fisher doing repeatability of the same sample, and you could see there's quite a variation. If you're dealing with, let's say, 3 or 2% of water content, sometimes you get very good parallel results, and sometimes in the replicates, there's a lot of variability, especially in the case I will draw your attention to, for example, in the 2.5 to 3 water percent range. It could be the difference between passing and failing. So we turn to near IR, and near IR is very good for SH, OH, NH, CH bonds. As shown here, we've used this for a number of processes that the chemists utilize in an open access format. They bring their vial in, they load it on the instrument, they collect the spectra, it applies them one of the many models we built, and they get the result typically under 2 minutes. I will draw your attention to the starred one, which is actually water, methyl THF, isopropyl acetate, and ethyl acetate. We collect one spectra. We apply multiple models to determine the concentration during this process. Now, this happens to be during a distillation solvent exchange going from one reaction to another, where they need to decrease one solvent, remove water, go to a second solvent to run the next reaction. And this is just a nice example here. We could pull out and develop these models for methyl THF, ethyl acetate, for example. We can collect just one spectra, apply it to multiple models, and get 4 results. That allows us to be able to avoid Carl Fisher and GC simultaneously. So in the biologics area, how about near IR? Near IR in water. We can do the determination of water in lyophilized proteins, whether it's an ADC or a monoclonal antibody. We first began utilizing one protein. We knew the Carl Fisher values. We do those after we collect the spectra. We can get very good correlation, but there's 6 factors used. It turns out that since near IR is temperature sensitive, it was best for us to allow the sample to reach the temperature at which it would be sampled at, where the spectra would be collected. So now the rule of thumb is don't leave your sample on the benchtop, put it on top of the instrument, which is equivalent to the diffuse reflectance mirror temperature. So we had very good results, but there's variability when you do replicates. We cleaned that up, we tightened it, we're down to 1 factor, and we could start adding proteins to our model. As a result, people do not want to do Carl Fisher anymore. They tend to rely on near IR, non-invasive, rapid, no sample preparation. Another advantage of near IR, we could differentiate the difference between bound water and unbound water. Shifting again here, we're using offline Raman spectroscopy. We use a 10x microscope objective. We can shoot inside of a jar. Usually we use clear borosilicate jars. We can use amber jars. There can be some interference from the amber, but good quality amber poses minimal interference due to its fluorescence or lack of fluorescence with good quality. And we also build models that avoid that broad spectral range that fluorescence can cause. On the left is an explosion-proof Raman instrument. We also have explosion-proof FTIRs and explosion-proof near IRs. On the right is where a probe is inserted in a sampling loop. It can be inserted directly into a probe as long as it's not behind a baffle, for example. You want a good sample mixing. We can also put this in a distillation line. So you could put it in a tank or in a distillation line post-condensate, so you could get an accurate sample during the condensation. What we do here then is we do Dynachem modeling. And in this particular example, we could use either Raman or near IR. We happen to choose Raman simply because we're going from acetonitrile and water, distilling it down and replacing it with isopropyl acetate. As long as they get the water down below 1% and the acetonitrile below 5%, they could proceed with their next step in the reaction. So they go through an initial distillation, then they go through a constant volume distillation where they're adding isopropyl acetate. We take multiple samples, we compare the results, and it turns out that all we need to do is put in a Raman probe at the distillate line, use Dynachem modeling, and collect the spectra, and we know when we are done with this step and we can proceed to the next step of the reaction. We tend to look at the quality of reagents. This is very important. You saw before using FTIR. In this particular case, as an example, we're using sodium methoxide and degraded sodium methoxide that the chemist had complained about. We obviously see difference in the spectra in both FTIR and in this example in Raman, good and bad. We go through a stability study. We take fresh, good quality sodium methoxide, expose it to air over 9 days, and you could see the breakdown occur over time. We see that there are multiple transitions, and eventually we end up forming some sodium carbonate. And this just shows some overlay of some spectra. You have your sodium methoxide, you have your bad sodium methoxide. You also have sodium formate and sodium carbonate all being formed. And if we want to, we could take that non-invasive probe, focus it on the top of a vial of good quality sodium methoxide, and watch it break down as it's exposed to typical lab conditions. If we move the probe to the bottom, we see it's still good quality material because the moisture hasn't penetrated through. And we could do fun things such as drying it completely in a balance oven, make sure it's nice and dry, then expose it to room air and just watch it decompose. All non-contact. And from this data, we could build a model. What else can we do? Replace titration. Not only replace Carl Fisher, but how about HCl in various solvents, such as methanol, ethanol, IPA. We could do that all by FT near IR. In some cases, we get very good results, such as potassium methoxide. We have models for potassium methoxide in ethanol by both Raman and FT near IR. Same thing with sodium ethoxide. We could use Raman or FT IR, near IR in ethanol. And lastly, I'd like to show the typical PAT approach. In this case, it's a telescope reaction. You're taking an aldehyde with ammonium hydroxide and methanol. It stays in methanol. You're using N-chlorosuccinimide or N-bromosuccinimide, forming a halogenated oxime. You do a coupling reaction, you get a product all in methanol. And for example, there you start with a clear solution, it turns into a slurry. We use the FTIR. We can see the decrease in the aldehyde during the process, the formation of the oxime. We can use multiple bands to watch the disappearance of the aldehyde, and sure enough, we could replace grab samples, and we took grab samples for comparison so we could build our chemometric models. And this also allows us to do other things, such as track the oxime formation and the disappearance of the aldehyde by changing the temperature, changing the agitation rate. This all sets up for production. Should we do a constant feed? Should we just add in bolus amounts ammonium hydroxide? So we could run those with either a probe or grab samples and take a look at the kinetics of the reaction. So, final lab tip. Thank you very much for your attention, and thank you, Spectroscopy Magazine.
Thank you, John, for that fascinating talk. As a reminder to everyone in the audience, you can submit a question by typing them in the Q&A box, which you can find underneath the media player. Also, please type in the name of the speaker your question is for. Questions will be answered at the end of today's session after all talks are complete. I would now like to introduce the next speaker, Julio C. Chacon Torres, PhD. He's an assistant professor and head of the nanotechnology department at Yache University in Ecuador. He earned his doctorate in experimental physics and material science from the University of Vienna, Austria, in 2013 with a thesis titled Theory in Spectroscopy on Functionalized Graphene and Graphite Intercalation Compounds. He also holds a PhD in nanoscience and nanotechnology from the Institute for Scientific and Technological Research of San Luis Potosi in Mexico, and a BS in Mechatronics Engineering from the Technology de Monterrey. Chacon Terras's research focuses on functionalized nanostructured materials for electronic and optoelectronic applications, as well as on the use of nanostructured clays as potential sustainable housing materials. He leads interdisciplinary collaborations across Germany, Austria, Hungary, Brazil, Ecuador, and Mexico. He supervises bachelor's and doctoral research, oversees laboratory design and equipment acquisition, and contributes to curriculum development in nanotechnology. He has also served as an invited international professor at multiple universities, lecturing in nanotechnology and material science. Dr. Chacon Torres actively participates in international conferences and serves as a peer reviewer for scientific journals. Julio, thank you for joining us today. You have the floor.
Hello, my name is Julio Chacon. I'm a professor at Yachay Tech University in Ecuador. That's a brand new university in South America with just 11 years old of being created. And I'm also the director of the Advanced Functional Nanomaterials Group, Anton. Today, I'm going to talk about Raman spectroscopy as a novel tool for tracing pharmaceuticals and biomaterials. So before to start, I want to show you quickly that our university is the first university that was created in Ecuador in order to develop science and technology. We are really proud about that because in 11 years, we have been able to consolidate in research, which actually is the reason why today I want to share these new results we have found. And during these 11 years, we have earned around 2,800 students. We are really small compared to other universities. We have 8 postgraduate programs, but we have been awarded 2 times with the Nature Index #1 in 2019 and 2020. Recently, 2025, we got the Nature Index #2. So we're really proud about this because even if we are a very young university, we have excelled on science. Our university is located at the top north part of Ecuador, as I said, in South America. But today, let's go to something more interesting that you want to hear, but you are always welcome to come and visit us at Yachay Tech in Ecuador. So today we're going to talk about how to detect low concentration of pharmaceuticals by using Raman spectroscopy. This is a really hot topic, I consider, because it has a lot of implications for science, technology, but in particular, to improve human health. Normally, it is very complicated since the pandemic to understand how does water in our rivers is being affected by the consumption of many pharmaceuticals like paracetamol, ibuprofen, that were 2 of the most used pharmaceuticals during the pandemic process, during the pandemic time, that we got a headache, and then during that time we had to solve it, or we got fever, and then we had to solve it, and then we were drinking pills. And at this time, you cannot resolve that problem by drinking just 1 pill, but instead using 2 of them or 3 of them. And the reason why is because we are getting resistant to these pharmaceuticals. The reason of that is because after we drink the pill, then your organism processes it, but not the 100% of that pill gets ingested and distributed along your body. Part of that pharmaceutical goes as a residue, and then you extract it from your organism, and then it goes directly to the water reservoirs. The problem is not that it goes there. The problem is that we cannot detect it in the water treatment plants. And these water treatment plants, they analyze different viruses, bacteria, E. coli, for example. But when they take a sample of this water, if you take a sample of that water and make an analysis for that, you won't detect the ibuprofen concentration or paracetamol because it's extremely low. So the idea of this article that you see on my screen is present to you a new way of studying the presence and the existence of these pharmaceuticals in a very low concentration regime. So this is what I'm going to show you today, and I hope you like this information. In order to start, first of all, we need to understand that pharmaceuticals like paracetamol and ibuprofen, they can be measured in powder, in a raw powder, by using Raman spectroscopy. It's a very straightforward, simple measurement where you simply put some powder below your Raman spectrometer, then you make the measurement, and you immediately obtain the fingerprint of this paracetamol or ibuprofen. That's normal, it's standard. And you get a spectra that is similar to the one that I'm showing you here, with all the vibrations that correspond to the different molecular sections that the whole molecule has. And if it has a carbon with hydrogen or carbon, oxygen, or amine or amide groups that are forming the whole paracetamol structure. You can confirm those vibrations by looking at the FTIR measurement, like the one we have here on letter C, okay? And then you get precisely all the vibrations that belongs to the structure, the molecular structure of these pharmaceuticals. That's fine, nothing new, nothing extremely rare. However, if you take a tiny amount of those pharmaceuticals and you mix them in water, for example, 100 milliliters of water, then you will notice that when you measure this water in a Raman spectrometer, you will see absolutely nothing but water. I mean, in principle, it's fine because the majority of that is water, though that makes sense. But if you think about it, I took a tiny amount of paracetamol, a tiny amount of ibuprofen, dilute it in water, mix it, and then you don't know that it's there because to our eyes, it's invisible. But, and also to the spectrometer, it's also invisible, but it is there. So I took it and I put it there, so I cannot lie. So how can we make our system more reliable so that we can really detect these tiny concentrations of paracetamol, ibuprofen, or any other possible molecule that could affect our biosystem? This was the main question we wanted to resolve here. So in order to do that, what we made is to design a system that could directly interact with the molecule of the pharmaceutical. So we took 0.01 grams of the pharmaceutical, we put it into 100 milliliters of water, and then we extracted only 2 microliters of that and make a tiny drop onto graphene on quartz, standard graphene on quartz. So we did that in order to determine if there was some interaction between the molecules, the pharmaceutical molecules, the structure of the molecule, and the graphene surface. And that was extremely interesting because what we found is that indeed the pharmaceutical makes a supramolecular interaction that connects with the graphene and then enhances the signal of graphene, and let us observe the fingerprint of our pharmaceutical molecule. Let me show you here our first result. Our first result was basically a graphene on quartz standard. You observe the D line, you observe the G line, the G line, the D line, no D line, no defects on our graphene surface, the G line, and the 2D line that is characteristic standard from graphene. After we deposited ibuprofen, that is the blue line that we have here, then we notice that the graphene G line signal was enhanced and the 2D line was much more enhanced. But when we put paracetamol, there was a stronger interaction that shows more than 10 times stronger signal on the G line and the 2D line of the graphene. That was amazing. That was really surprising us because it really tells us that there is something happening between the graphene structure and the molecule that it's been deposited on top of the surface of graphene. Moreover, we notice here some additional peaks that appear after we deposited the molecule of the pharmaceutical on the graphene. For example, those peaks around 3000 and some additional peaks in between the D and the G line and also on the low frequency range below the D line, okay? Those peaks we directly correlated to the molecule that we were depositing. So what we did was a deconvolution analysis of the Raman spectra, obviously first of the pristine graphene G line. So we notice some of the peaks of quartz because it's below, underneath of the graphene, and the G line is clean, nothing else. But after we deposited the ibuprofen, we noticed that there were some additional peaks and also on the paracetamol. And these additional tiny peaks, they were related to the presence of the molecular vibrations of our pharmaceutical. We actually compared the positions of those peaks to the ones of the pristine material, and we confirmed that indeed they belong to this molecular structure. But then, one of the questions that was asked to us is that if there is a direct dependence of this molecular fingerprint compared to the concentration of the pharmaceutical that we have deposited. So indeed, there is one, okay? So what we did is to dissolve different amounts of the pharmaceutical into these 200 milliliters of water. So what we did is to make a 10 micrograms per milliliter concentration, 20, 30, 40, 50, 60, up to 100 micrograms per milliliter solutions. We observed an evolution of the UVB spectra, which was expected as a function of concentration. And then if we focus that the main peak of excitation for the UVBs is located around 222 nanometers, then we excited the sample up at this energy, and then we analyze the absorbance at that particular energy and make a calibration curve that tells us that the concentration is dependent, the absorbance is dependent on the concentration that we have. This is just a standard in order to know that indeed we have a dependence on the concentration. Sorry. What happened when we make the analysis of the Raman spectra? When we make the analysis of the Raman spectra, our pristine graphene is the lower peak that we observe here. It's not so clear because we overpose one on top of the other, but we immediately observe that additional peaks arise not only on the DG region, but also on the 2D region, and they are extremely strong and clear to be observed. We immediately assign those peaks on the high frequency region to the CH2 and CH3 molecular vibrations of the molecule that we were studying, in this case, ibuprofen for this graph, okay? And after that, we perform a three-dimensional water plot, in this case, to determine how strong was the enhancement of the signal with this deposition, and we observe that the strongest signal was obtained around 20 particles per million that are present in our solution. Why not on the strongest concentration, like 90 particles per million, ppms? Because if you think about it, after you deposited a higher concentration of molecules on top of graphene, that will provoke that we get a thicker layer of pharmaceutical, and then there is not a direct connection and a direct interaction between the molecules and the graphene. But with a lower concentration, we have a thinner layer of pharmaceutical molecules, and those are directly interacting and creating a hybridization between the graphene substrate and the molecule. So we consider that possibility, and then we make again our deconvolution analysis, and then we obtain the peaks that are related to the CH2, CH3 molecular vibrations of the molecule, and in addition, all the fingerprint vibrations that are corresponding, in this case, to ibuprofen, as we observe here. So are very clear here, and we actually corroborate those positions that we observe here on this compound that is deposited on graphene to the pristine vibrations. And in this case, what we observe is that indeed they correspond and they are exactly aligned to the ones that we obtained from the pristine powder, where we just had ibuprofen or paracetamol. In that case, our main vibrations are located on the rocking vibrations or carbon-carbon stretching modes, or the CH symmetric stretching vibrations, or carbon NH deformation vibrations that we could observe in paracetamol in particular that are not on ibuprofen. So we can really distinguish between the presence of ibuprofen and paracetamol in these two cases. And what is more exciting is that for the first time, at least to my knowledge, okay, we have a really well description of the fingerprint of paracetamol or ibuprofen, these two pharmaceuticals, in an extremely low concentration. A concentration that cannot be easily detected when you have the whole liquid system, but when you deposit it on graphene, then you look at it and then you have the signal appearing there. So we did that system, but we wanted to be sure that the response was really coming from an interaction between the graphene and the molecule of ibuprofen or paracetamol. Then we make a map analysis from the place where we deposited the drop. Not the whole drop, but a section of it, as you can see, several microns of distance. And then we observe that in the region where the drop was deposited, our signal for graphene was stronger when compared to the area where there is just graphene on quartz. like here, where there is just graphene on quartz, then we observe that there is no signal for the D band, G line, nor the 2D line, okay? Or it's much more smaller when compared to these other areas. That's why it looks black, okay? But these other areas are way stronger and they got thousands of counts, and we can really say that there is a strong interaction between the molecule of the pharmaceutical and the graphene surface. And that can tell us directly in an immediate way that we have something, a molecule that it's coming from our pharmaceutical, because there is nothing else but that, and it's immediately interacting, and there exists a hybridization and molecular interaction between the pharmaceutical molecule and the graphene lattice. That it's not being destroyed because the D line is not increasing, but instead it's kind of a functionalization created between the molecules. Then we decided to study something else before we could finish the experiment, and it's the conductance of this graphene. We know that the graphene conductivity is very important, and we wanted to know if it was affected by the presence of the molecule, and indeed it was. So we wanted to remove the pharmaceutical by heating the quartz plate, and then the graphene got oxidized. And then now you see that the spectra doesn't look anymore like graphene. It looks more like a graphene oxide, still with some structure. But when we made the 4-probe measurement analysis, our material was not with the expected conductance of 25 kilosmers per centimeter, but instead it changed into 3.5, between 3.5 and 5, which actually is way lower, but we attribute this effect to the functionalization, covalent functionalization sites that are created between the graphene lattice and the molecule that it's been absorbed within the surface of graphene. So with this information, we were really happy because for the first time, we are able to trace these supramolecular interactions between graphene and pharmaceuticals. And we know that the pharmaceutical triggered the graphene surface enhanced Raman response, which is really interesting because it can give us the possibility to detect pharmaceuticals in low concentrations to the limit of 100 parts per milliliter, observing a strong Hertz enhancement for the G and the 2D line up to 48 times stronger, okay? So that was our conclusion, and I want to invite you to participate in our 6 nanoscience summer school that it's going to be held in Galapagos Islands next year. It's already you can access all the information and register in the QR information that we have here. With this, I want to thank all my group of students. They already graduated, but Nigeli and Nardi are the two who were really working hard on the development of this system for the detection of pharmaceuticals. Thank you very much for your attention, and I'm going to be open for questions.
Thank you, Julio, for that excellent talk. And thank you to all of our speakers who have taken the time to present during today's morning session. As a reminder to everyone in the audience, you can submit a question by typing it in the Q&A box, which you can find at the bottom of the screen. Once again, please type in the name of the speaker your question is for. It is now time to begin the question and answer session with today's speakers. So let's start with the questions. Well, thank you, Julio, for that excellent talk. And thank you to all of our speakers who have taken the time to present during today's morning session. As a reminder to everyone in the audience, you can submit a question by typing it in the Q&A box, which you can find at the bottom of the screen. Once again, please type in the name of the speaker your question is for. It's now time to begin the question and answer session with today's speakers. So let's start with the questions. This first one is for Igor. First of all, the question asker says, "Thank you for the excellent talk. I was wondering whether the samples were in liquid form, and if so, was the system a macroscopic or microscopic system? Also, do you know why are lipid signals so low in the Moderna nanoparticles when typically lipid signals from tissue or other samples are quite strong?"
Well, Catherine, first thank you for your favorable comments and for very important question. The system which we used for this study is home built. It's not a microscope. We collected scattered light with single lens with high numerical aperture, and coupled the collected radiation into double spectrograph. You are absolutely right that we may expect typically stronger signal from lipids, but you see, we specifically chose conditions from the viewpoint of accumulation time that this signal is low, because we specifically wanted to measure signal from mRNA. And we used 266 nanometer excitation, which allowed us to get resonance enhancement from mRNA. As a result, a signal from lipids was low. And the second part of your question. Oh, no, I think that's it, right? Thank you.
Yes. Next question is for Igor. What is your explanation of quite different spectral changes caused by two different mRNA degradation experiments? I mean, why a significant decrease in the Raman band's intensity is observed only after degradation with RNase A?
Yeah, this is good observation in our slides. I didn't talk about it in details, but I believe that RNA's degradation resulted in more significant changes in the sample. I believe that we may see some precipitation in the sample because of this phenomenon. However, what was important for us is that under both conditions, under both types of degradation, we saw similar effect on Raman band shifting.
The next question is for Igor as well. Have you verified whether irradiation of mRNA vaccines with deep UV light could damage the mRNA and render the vaccine non-viable?
Again, very important question for practical application. We only verified that irradiation with 266 nanometer laser doesn't change any spectral property. We didn't check mRNA biofunctional activity yet, but this is something we will do in the future. Thank you.
So Igor, will your in situ stability test of commercial mRNA vaccines require special containers, since typical vials are not transparent in deep UV spectral range?
Correct. Yes, again, practically very important question. At the moment, vaccine containers are typically made either from glass or some plastic, which is not UV transparent. However, there are plenty of plastic and polymer materials which are transparent in UV region, specifically
around 250 nanometers. So I believe that it will be possible to make containers which are suitable for vaccine usage, and at the same time those containers would be transparent in UV. Thank you.
This question is for Joseph. How do you go about evaluating inline versus online versus at-line PAT tools?
Thanks for the question. It's a really important question, and I would say from the talk, some of my thoughts on this are we try whenever possible to prioritize inline over online over at-line tools. Again, just to level set a little bit on what that means. Inline tools, how we think about that is analytical instrumentation and technologies that directly interfaces with a process itself. So that might be a Raman probe that sits within a bioreactor. Online tools are things where you need to flow from the process to the instrument, and then you either flow back to the process or flow to waste. And then at-line tools are essentially when you need to sample, for instance, a reactor, a bioreactor, and then bring that sample to the analytical instrumentation. If that's close to the process, that's usually turned at-line. If it's further away from the process, that's usually turned offline. Some of the reasons behind prioritizing inline over online over at-line were kind of showcased in the talk, but I think to us, it's really trying to get as real-time analytics as possible and as representative analytics as possible, meaning we see PAT tools as providing us an opportunity to have eyes into the process, so to speak. So if we're able to have analytical instrumentation where we're measuring phenomenon in real time, in situ, directly within the process, we feel like that's as representative as possible. Obviously, all tools can't be inline necessarily, so there's value in tools across the spectrum of inline, online, or at-line. But I think for us, we try to evaluate tools that will directly interface with a process whenever possible and kind of go through that line of priority thereafter.
This is also for Joseph. What are some key considerations when developing a process analytical technology or PAT method to enable pharmaceutical process development?
Yeah, it's a really good question. Some of the linking to the previous answer, I think one, it starts with what is the problem statement? So a lot of the times, right, are we trying to understand our process better during development? If some of those are questions that we're having, maybe we take an exploratory approach of, like I mentioned in the talk, kind of quote unquote throwing the kitchen sink at a process. We have a lot of tools. We have microscopy, spectroscopy, et cetera, et cetera. Can we look at kind of exploring PAT evaluations to understand more about the process? You know, on the other hand, if the problem statement is more, hey, we have a specific analyte of interest, maybe we have a nutrient or a metabolite that we would really like to measure and quantitate in real time to be able to control a process off of, for instance, then we take a more targeted and uniform, kind of unified approach, so to speak, of, hey, what's the best technology to measure this analyte of interest? How do we go about doing that? Would there be inline technologies where we could do this in real time? Would it be online? You know, would it be at line? Things of that nature. But I think to me, what's really important is first starting with what is the problem statement? What are we trying to do? Is a PAT method the solution, or is just another analytical technique the solution? And again, when you hear problem statements that deal with deeper understanding, representative analysis, inline, real-time, process control, things of that nature, usually that's kind of where we like to focus our PAT method development. The only other thing that I'll mention with this is that I think for us, data collection and data analysis really go hand in hand. So sometimes folks will focus on one end or the other, but I think for us, it's when we're developing a PAT method for pharmaceutical process development it's what is our best data collection strategy, and then what is our best data analysis strategy. And I think I just wanted to mention kind of the intersection of a lot of the data science tools with the measurement technologies is I think the sweet spot, and a lot of opportunity for more PAT method development and deployment across pharmaceutical process development as we kind of lean into that intersection.
And for Joseph, how do you approach exploratory PAT tools?
Yeah, it's a great question. I feel like for me, it's a really, really important endeavor where when we're looking at process analytical technology, we have kind of bona fide, tried and true technologies out there. For instance, Raman spectroscopy, right? It's been used across the industry in a variety of different settings. It gives really rich information. The ability to partner Raman spectroscopy with chemometric and multivariate approaches can quantitate really important metabolites, nutrients, things of that nature. There's also the other end, right, of as we're developing new technologies, how do we, like I mentioned before, kind of throw the kitchen sink at things? There are things like I showcased in the presentation. We have microscopy techniques in situ microscopy where maybe there's challenges when it comes to autoclavability or aseptic connections that might at this stage limit practical implementation across every single setting in a pharmaceutical environment. But to me, when we're exploring those and saying, okay, we're finding the right problem statement, we're finding the right opportunity, we're going to explore PAT tools that have maybe not been used to date, but we think there's a lot of value in that particular problem statement. Once we get kind of really important case studies and data that showcases, for instance, hey, inline microscopy tools are going to be really useful, then we can perhaps help influence, turning and transitioning those type of PAT tools into things that can be autoclavable, that can be aseptically connected, for instance, to kind of have a more long-term strategy in terms of implementation. But to me, it all starts again with a problem statement, right? Do we have tools that maybe haven't been historically used that we can try, that we can explore? It's science, so sometimes stuff's going to work out, sometimes stuff isn't, but I think there's a ton of value in exploring new approaches, and then once things work, kind of look for more long-term implementation strategies.
And for Joseph, what are some important factors for getting new technologies adopted within pharmaceutical process development?
Yeah, it's a great question. I would say some of the important factors, like I mentioned, are, once you've explored an approach and you have a really good, for instance, proof of concept of, hey, we might have a new process analytical technology that we want to implement. I think a lot of things come towards a practical implementation setting of, hey, if this is an inline tool, how are we going to interface this technology with the process itself, for instance, a bioreactor, right? So a lot of things come into play there where we might actually be doing a lot of custom automation or various things of that nature to be able to physically integrate hardware. We also need to digitally integrate systems and hardware as well. So it starts with an analytical method, but then in terms of getting things practically implemented in a pharmaceutical process development setting, I would say it's augmented from just the analytical side of a lot of engineering tasks, a lot of custom automation, a lot of systems and hardware integration to be able to say, hey, we've now solved a lot of practical challenges with getting this probe-based Raman spectroscopy tool, for instance, into a novel bioreactor. Things of that nature, I think, are really important. The other thing that I would say too, and I mentioned this in the talk, something that's really important to us at Merck, especially in the data-rich experimentation team, is really building trusted partnerships across the team. So let's say our partner is responsible for running that bioreactor. We need to really understand, if we're trying to look for implementing new technologies into that setting, what are the problem statements? What are the workflows? How is that done currently? And then I think once we understand and kind of build a trusted partnership to do that, then we can make sure we're solving the right problem, right? It might turn out, for instance, hey, an at-line tool is actually exactly what we need for this, problem statement or this challenge. So in those cases, we wouldn't necessarily look for inline tools for implementation. So I think to me, it all stems from understanding the problem statement, understanding your partner needs, and then once you kind of build that deep partnership across, hey, process development, technology development, that's really where a lot of opportunities exist.
Well, thank you, Joseph.
Thanks.
Here are some questions for John. What is your typical approach to deciding which instrument is the most suitable for a particular analysis?
Okay, can everyone hear me?
Yes.
Thumbs up. Great. We typically, when challenged with a question from some of our synthetic chemists or the biochemists, we quickly pre-screen. And two approaches. One, they can actually walk up to an open access instrument and take a spectra and we can evaluate it, or they can evaluate it, or they give us samples. If it's something that we can get a hold of a beginning of a reaction and end of a reaction when they're screening, we can either look through our library to see what has been collected in the past, or quickly do a fast scan using FTIR, near IR, fluorescence, UV. We run the gamut, and we have these instruments available as open access. So anybody can use them, or they can bring the samples to us. We pre-screen and see which is best. And as you know from chemometric model building, the longest time is actually prepping your samples, especially if it's going to be GMP, to make sure you have good samples to collect the spectra, to begin to build your model, to add to some sort of a process. So that's our usual steps. We also base it on knowledge. Take a look at the molecule, understand what have I experienced in the past, and then we can hone in on it. But sometimes there's always surprises, and there's always interferences too. So we like to have the best and worst case scenarios when we try to analyze.
Well, here's a follow-up question, John. How difficult is it to transfer methods from one instrument to another if they are from the same instrument company?
From the same instrument company, we're actually very, very lucky. It depends on the approach. For example, with near IR, the instrument we typically use has internal NIST standard light grading polystyrene. So whenever the light source is changed once a year, and you do have to pay for it, but it ensures that they match a NIST standard, and we can do a transfer from one instrument to another. In some of the other cases, we do a lot of normalization prior to building a chemometric model, prior to doing, for example, a PLS model, normalized to a solvent or something constant in it that's going to vary in production. Then, because if you're charging in a large scale, sometimes it's 1 kilo, 1.1 kilo, 1.03 kilos, there's variation, but we normalize the spectra to another component in that spectra. And then we build our models. And so that has helped ensure us being able to transfer from instrument to instrument.
So following along...
I'll also add, we do something called co-validation. And that's actually very fascinating. We've been very lucky with it, where we will actually validate a method, send it to another location, whether it's in the US or abroad. They run new samples, collect the spectra, send the new spectra to us. We, in turn, collect brand new samples, collect the spectra, send the spectra to them, they process it. And if both sets of numbers are identical when we exchange the raw spectra, then we can ensure that we're going to get the same results. And that helps in our co-validation. The bad part is it keeps us from having to travel to exotic sites.
As a follow-up, how difficult is it to transfer methods from one instrument to another when they're from different manufacturers?
That's a challenge we've had, and we've been fortunate because we've been able to take some of our models that are like MATLAB based, rework the models with the raw data because we have the actual raw spectra, and then rebuild the models or adjust the models so that they work. It's a much more of a challenge. That's why we tend to hone in on a certain vendor's instruments that have robustness, have everything we need, and then we would purchase series of them.
Well, is it easy to validate a method? What are the challenges in validation?
Well, reproducibility, for example, accuracy and precision. We try to follow the traditional HPLC requirements as close as we can in order to do an accurate validation. And that the point I mentioned about the co-validation, that's where spectroscopy actually helps us because there's too many variables, added variables in HPLC or GC, the length of your tubing, the quality of the packing material. Even if it's the same C18 or cyano column, there's still going to be variabilities, let alone the diode array or fixed wavelength. there's going to be strength, intensity differences, and that's going to also impact you. So this is why we try to match the instruments. We do a lot of pre-checking of the instruments to make sure they're aligned in terms of, for example, laser power, throughput. And that means checking your laser output not only at your source, but at the terminal end of your probe, whether it's non-contact or contact. And that's a pre-check to make sure you're aligned when you transfer methods.
And John, are most instruments capable of interfacing with the SCADA system?
Yes, yeah, we've actually worked quite heavily with different SCADA systems so that we can have this supervisory control and data acquisition. Many times we tie things into, for example, our Delta V system. So we have control in Delta V. That way the operators can see the results, especially if it's live, if it's a true PAT endeavor. They can get the accurate results showing up on the screen as it's occurring, because obviously when you're in production, you want to work as quickly as you can. And we've had examples where if you let the reaction run too long, then it begins to break down. You get side products, you lose your yield. So interfacing, I think, is very important for process control, for making adjustments on the fly and understanding when to terminate a process.
Thank you, John. So here's a series of questions for Julio. What response do you expect of having when using different laser excitation wavelengths?
Thank you so much for inviting me. And well, regarding that analysis, it will really affect the fluorescence response from the material. However, one of the advantages of this system is that when we have graphene on the underneath of the molecule that we are depositing, then the fluorescence is being quenched by the graphene. And normally we will get the spectra from the Raman response of the main molecule that we are analyzing, on top of the graphene Raman response. The thing here is that obviously some of the Raman vibrations are not active when we go to the infrared spectra or to the red ones. So then in that case, when we use lasers that are lower energy, then we will have less response from the graphene, but a higher intensity coming up from the molecule that we are analyzing.
And Julio, have you tried different pharmaceuticals on the same system?
Yes, we did. So in the publication that we presented here during the talk, we have shown the ibuprofen and paracetamol response on the graphene surface. However, we have tried also 3 different more materials like ampicillin. We have tried amoxicillin. And in this case, we observed that the enhancement is not as high as the one we obtained from ibuprofen and paracetamol. This may have to do with the fact that that there is a correlation between the substrate that we are using underneath of graphene and the response of the electrostatic interaction between the molecule that is sitting on graphene and the substrate underneath. So we have noticed that there is a strong dependence that we are now evaluating when we have graphene on copper, when we have graphene on silicon dioxide, when we have graphene suspended even. And then there is a strong dependence depending on the type of molecule we want to analyze. So it's kind of getting a selective method for different type of pharmaceuticals.
And Julio, what is the importance of using graphene as a template for the surface-enhanced Raman spectroscopy response in your system?
Well, normally on a search system, everyone is using nanoparticles, such as gold nanoparticles or silver nanoparticles, in order to induce this search effect on molecular detection. But now, when we have a flat system, a flat surface like graphene, then we are not limited for the position, the location of the nanoparticles on the substrate. And that gives us a huge advantage, because then on a small 1 × 1 centimeter square system of graphene, then we are able to deposit several drops of different molecules, and then all of them should give us kind of the same signal because it's a flat homogeneous system that is not strongly dependent on the shape and the position of the nanoparticles per se, that are actually the reason why the surge effect gets enhanced or quenched at the same time.
This is again for Julio. It says surface-enhanced Raman spectroscopy suffers from reproducibility. Do you see reproducible data between repeated graphene quartz substrate preparations?
Yes, indeed. So this comes actually from the previous answer. So one of the problems for the reproducibility for the SER systems is the shape and the distances between the nanoparticles in order to induce this enhancement factor. However, when we have graphene, as it is a flat layer on top of the quartz substrate, then we have tried and we have tested different points at different positions of the graphene lattice, and then we obtain the same result. There is a dependence only to the presence of the molecule we want to study. If we are measuring outside of the molecule, then we don't get the Hertz enhancement, the one that it's induced by the graphene. But when we are on the molecule that is sitting on graphene, then we get this Hertz enhancement, and it's the same as if we measure here or 1 millimeter above or beyond the first one we have measured. Also, something I can say is that the first measurements that we present, I think in the second figure of the paper, those measurements were done at first, and then afterwards, during the review process, the referee asked us to repeat the experiment as a function of concentration. That is something I mentioned during the talk. And at that point, we had to make again a new sample to make this process as a function of concentration, and then we observe a reproducible process. So that was actually a really good thing from the referees, because it forced us to repeat the experiment from scratch and going from 0 to high concentration. And then we really observe the trend of the change of this system. And this is something constant. So it's not something that happened just once in a moment, but something that you can really reproduce.
For Julio, another question. Graphene enhanced Raman has great sensitivity. How is its repeatability?
Well, it goes online with the previous question. So graphene enhanced Raman, it's something I would say recently been studied more than before. And I like it very much because, as I said, it's a flat system that it's very reproducible in that sense. So it doesn't depend on the size of the lattice. It only depends on having graphene really attached to a flat surface of either quartz, or in this case, what we are recently studying is graphene and copper, graphene suspended, and graphene on silicon dioxide. But repeatability of this experiment has been proved to be quite consistent. So we like it very much.
This is for everyone. Thank you to all of you for your presentations. The next question is for Joseph. What are the major challenges with scaling up PAT from the R&D stage to manufacturing?
Yeah, it's a really good question. And I would say some of the things that we consider are, one, not every PAT tool needs to be scaled to manufacturing. So some of the examples like showcased in the presentation were during process development, we uncovered some clumping phenomena that maybe we missed and didn't have a deep understanding of from just offline analytics solely. So because we noticed that during development using novel PAT tools, we were able to actively mitigate, correct, solve that challenge. So now we don't actually need PAT in a manufacturing environment because that's not actually a challenge anymore, because we kind of invested in PAT at the process development stage. So I think the first thing is you have to be selective on what is actually critical for the program to be successful as it moves through the development stages of the pipeline. And once you have that selection, how is PAT going to specifically enable that? certain things, like for instance, process control strategies, those are going to be most of the time a entirety of the life cycle of the program. So if during development we say, hey, Raman spectroscopy can be useful for measuring these metabolites and nutrients, and we want to actually control our process based off of that, that would be something that we would definitely prioritize to continue to, as we scale up the process, for instance. We want to make sure that we have Raman spectroscopy embedded in that to be able to control the process effectively. And to me, I think, seeing the evolution of the PAT field in general, going from a purely manufacturing focus with PAT tools to now shifting gears towards more the entirety of the development stages, especially during development, I think is an exciting time where we might be able to uncover deeper understanding about the process that we can use selectively as PAT tools as the program progresses, or we have things that are kind of mission business critical to say, hey, we want to control our process based off this PAT. So therefore, through the entirety of our commercial manufacturing, we're going to make sure that this PAT tool is implemented.
Well, thank you. This is for any or all of our invited speakers. It says partial least squares is the de facto standard for regression analysis, but is it possible that machine learning approaches will be adopted in the future instead? Does anyone want to take that?
I'll take a stab at it.
All right.
Because I think we have tried some kind of machine learning to develop predicted spectra. It's very difficult with near IR. It's much more easier with a nice fingerprint like FTIR and Raman. We come up with something unusual. We try to piece together what the functional groups might be and then work with software to give a predictive spectra on it based on library values, known compounds, etc. It's obviously a challenge to be able to do that. I imagine once the libraries are more set up, fully established, the predictability using AI would help. And that may also lead to how you would select which instrument might be the best, because you can have so many different tools. There would give you an indication of using detector X versus detector Y, or instrument X versus X, Y, or Z to get the most reliable spectra. But the caveat to that is if you're in a complex matrix, yeah, AI has to do a lot of work.
Well, thank you, John. This is also for any of our invited speakers. UV light itself can cause degradation of sensitive molecules, including nucleic acid and protein. How do you address that in any experiments?
I guess I can tell a little about this. So we have been using deep ultraviolet light for generating Raman scattering from biological systems for a long time. And it was a big concern in the beginning that deep UV light will damage our samples. And for this reason, we developed special cell which would allow either mixing or even using a constant flow system for the measurements. At the same time, what we did, we measured quantum yield of photodegradation, for example, for proteins. And we found that from the viewpoint of changes in spectra, meaning that you make changes in chemical composition of the sample, quantum yield is very low. In fact, proteins are quite resistant to any significant changes due to deep UV radiation. And when I say deep UV, we really use deep UV. We use the radiation below 200 nanometers. However, we haven't tested physiological activity of proteins, and I think I mentioned it already earlier in this discussion, that this is something we may need to do if we go to practical application, for example, for using deep UV for testing testing the stability of mRNA vaccine or any therapeutics. But my general impression that deep UV doesn't make much changes in biological systems. Thank you.
Thank you, Igor. This is also for any of our invited speakers. How do you deal with the issue of instrument wavelength or wave number drift over experimental periods?
Well, this is a general question. I think everyone can comment on it. I probably start commenting on it because we are using home-built instrument, which of course not as robust as commercial instruments. And I mean, in order to check that there is no any shift in alignment, of course, what we do, we run control samples with frequency which we think are useful for specific experiment, right? The worst case scenario, we run, for example, sample of Teflon after each measurement of our real sample. And typically we don't see much changes, but once in a while we do see changes, and then we practically need to make realignment and repeat the experiment. Thank you.
Well, I want to comment on that quickly as well. So in the case of a Raman spectrometer, normally what we use is a helium or neon lamp. In this case, we perform a measurement on that before, after, or during the measurement, so that we get the peaks from neon, for instance, and then we calibrate on that. So because if the person who made this question notice a complete shift of the whole spectra, that means a calibration error. So then it's necessarily that you perform a measurement with the standard so that you get the shift, and then you can calibrate the spectra. Because in the case of a Raman spectroscopy, then if you get a shift of 3 to 5 wave numbers, it may mean some doping effects, strain effects, additional effects that are induced into your sample. And if you do not correct this calibration, you may mislead the answer and the response of the sample that you have there. So it's very important that you perform this calibration process.
Well, with that question, let's wrap up this session. We want to thank all the invited speakers for your presentations, which were very excellent. We want to thank the audience for attending and for participating in the first morning session of this virtual symposium, Emerging Trends in Pharmaceutical and Biopharmaceutical Analysis, presented by Spectroscopy, the Society for Applied Spectroscopy, and the Koblenz Society. Thank you again to our sponsors, CEM, PerkinElmer, Thermo Fisher, Metrome, and Andreessen Hauser. We also want to remind you to take advantage of the resources from our sponsors, which you can find on the bottom of your screen. You can still sign up for the afternoon session in this virtual symposium using the link in that resource section. If you would like a certificate of attendance for this event, please send an email within a week of the event to training@sas.org. That is training@sas.org. You will receive an email alerting you when this virtual symposium is available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. We'll now break for lunch ahead of our afternoon session. We hope you'll join us as we reconvene at 2 p.m. Eastern Time, where we'll hear leading instrument suppliers give educational 20-minute talks addressing application tips, the best practices in the use of vibrational spectroscopy, and more. We also hope to see you at our other sessions where we have more great lineups. Goodbye.